All right, this is a really quick podcast just comparing prokaryotic and eukaryotic DNA, its differences, and how those differences can impact how prokaryotes and eukaryotes copy their DNA or replicate their DNA. Prokaryotic DNA is much more simple than eukaryotic DNA. It is a circular molecule, so this is representing the bacterial DNA molecule. It would be a double helix, just like eukaryotic DNA with complementary nucleotides, but it is arranged in a circle and connected, so this end would be connected to that end, and it would just be in a circle. It is only one strand, plus some little accessory DNA molecules called plasmids, that are important to bacteria because they can carry genes that confer antibiotic resistance or something like that. The main genes required to allow the bacteria to function are in the main prokaryotic chromosome, the big circle. But there are smaller, tinier pieces called plasmids that are important because they carry extra genes, and also we now use them in our own genetic manipulations of organisms in genetic engineering. We have learned to use plasmids to our own advantage, which we will talk about later. Also, prokaryotic DNA is naked, meaning that it is not associated with any other molecule except this DNA strand. And eukaryotic DNA is different from that. First of all, there's lots more DNA in a eukaryote than in a prokaryote. So there is less DNA in a prokaryote. Eukaryotes have more genetic information. It is arranged in linear molecules, so the classic DNA double helix that we are used to seeing in a straight line, that would be a eukaryotic DNA strand we would call a chromosome, okay? Whereas again, the bacterial DNA comes in this circular double helix shape. Okay, there are multiple pieces of DNA strands, multiple strands, multiple chromosomes in each eukaryotic cell. If you are a human, you have 46 pieces of DNA or 46 chromosomes in your cells. Whereas if you're a bacterial cell, you only ever have this one piece. Also, in eukaryotic DNA, the chromosome is not just made of DNA. It is not just made of DNA. It is DNA that is coiled around proteins. These proteins are called, and you need to know this or recognize it, histone, that would be an H, histone proteins. And these proteins act, well, if you have a thread, if you go to a store to buy thread, they don't just sell you a pile of thread. It is wound around a spool so that it won't get tangled up. Well, consider a similar case for DNA. We don't want this massively long DNA molecule to get tangled up, so we coil it around these proteins that act like spools for us. The DNA coupled with these histone proteins forms a complex that we call chromatin, and when the chromatin gets really thick during cell division, we can see the individual pieces as chromosomes. Okay, so if we look at this is kind of showing some detail about how chromosomes are structured in a eukaryote. So here's our DNA, which is our A, C's, T's, and G's, and the double helix right here. But we wind that double helix, seen as the pink line here, around these spools, and these spools are our histone proteins, okay? And each DNA and spool complex, each of these is called a nucleosome. I'll just write that so in case you see it, not a nucleus, but a nucleosome, a very tiny ball of DNA and histone protein. But these balls of histone protein and coiled DNA are then coiled on top of each other, as you can see here, and then loops are made of all these coils of histone and DNA, and then loops on the loops on top of the loops, and then that's how you get this very densely packed, incredibly densely packed DNA molecule we recognize as a chromosome. Okay? All right. Now, when copying DNA, we've already talked about an origin of replication, where the replicating starts. In a bacterial chromosome, prokaryote, there is only one origin of replication. Okay? It starts replicating, say, here, and it's going to proceed in both directions like this until they both meet, say, over here, and then you end up with two circular chromosomes. Okay? And a eukaryote, if you'll go back here, they have multiple origins, mostly because there's so much more DNA in a eukaryote than a prokaryote. If you only started it at one place, if this is my eukaryotic chromosome, and I started copying it here, and I had to go all the way to the end of it, it would take me a very long time. Whereas if I start in multiple places along the strand and then I just have to meet in the middle, then I can cut my time dramatically shorter that I need to copy two entire strands of DNA. So multiple origins of replication make replication faster in eukaryotes than it would be otherwise. Okay? All right. By the way, here you can see the loops, the origins of replication. Here is one. The fork would be right here, and we're replicating this way. The fork is here. We're replicating this way. Here is another loop. Here is another loop or origin here. So that's what it looks like in real life. All right. Thank you for watching, and that's it. And I'll see you in the next podcast. Take care.